Started. Started here. Started. పెట్టిపోయినా పెట్టాను. Thank you. Okay. So, in continuation with the third to eighth week development, we are dealing with some special points which are important to deal with. So among them, now we deal with the amnion, yolk sac, allantois, chorion, decidua, and umbilical cord. So the first one is the amnion. Amnion is thin extra-embryonic membrane that loosely envelops the embryo, forming an amniotic sac that is filled with amniotic fluid. Amniotic sac is lined by amnion is a thin extra-embryonic membrane. So we are talking about the membrane only that loosely envelops the embryo, forming an amniotic sac that is filled with amniotic fluid. The amniotic sac is lined by ectodermal cells of the inner cell mass and amniogenic cells of trophoblast. So this amniotic sac is lined by ectodermal cells of inner cell mass and amniogenic cells of trophoblast. Amniogenic cells line the roof and lateral walls of the amniotic cavity and floor is formed by the ectodermal cells. As amniotic sac enlarges during late embryonic period due to collection of more amniotic fluid within it, the amnion gradually surrounds the whole embryo and also covers the developing umbilical cord. So here, if you remember properly, that is in the second and third week of development, wherein the embryo is traveling from the ampulla region to the uterine cavity. So while it is traveling, it is, from morphular stage, it is collecting fluid, and inside you will be seeing the separation of embryoblast and trophoblast, and the trophoblast in the embryoblast we will be seeing the development of epiblast and hypoblast, and then in epiblast the cells gradually are collecting fluid and then they are separating to the center, and then you are seeing the amniotic cavity formed. So now this amniotic cavity is formed here. This is the amniotic cavity formed in the blastocyst, and this amniotic cavity is surrounded by a layer called amniogenic layer, which is derived from the trophoblast, central trophoblast that is surrounding all of the embryo. Then on the other side there is an endoderm, and the space below the endoderm we are calling as primary yolk sac, and flat cells develop from endoderm and cover the inner layer of the trophoblast in the primary yolk sac, which we are calling Husser's membrane. And then of course the trophoblast develops gradually the mesodermal cells, into which the cavity develops, coelom we call it as, and then these mesodermal cells cover both the yolk sac and amnion, and then this will go into the stalk, connecting stalk. So this is how the amnion is a cavity, small cavity first, then yolk sac is there, and in between we are having embryo plate, and then this amnion along with embryo plate is folding on the median line, and then as it folds it is absorbing part of yolk sac inside this fold. So finally we are seeing the primitive gut is formed from the yolk sac that is absorbed into the folds of this amniotic cavity or the embryo itself, you can say, and then a part of the yolk sac is remaining. This will talk again. So now the amniotic cavity grows at the expense of extra embryonic coelom, which gets obliterated and fusion occurs between amnion and chorion. So, as the amniotic cavity enlarges, it first pulls the embryo, covers the embryo, then covers whole of the yolk sac as well as the amniotic sac, such that it is almost, touching the coelomic cavity, completely covering the coelomic cavity. Then, as the as it grows more, the trophoblast layer and the inner layer of this outer layer of the coelomic cavity are coming together, such that the chorion, the trophoblast layer is nothing but chorion and amnion are coming close together, so that the coelomic cavity is disappearing completely. So now amnion consisting of two layers, outer layer made up of somatopluric layer of extra amniotic mesoderm and inner layer amniogenic cells derived from the trophoblast. This is still in the amniotic cavity, not fully developed. Now amniotic fluid, it is a clear water fluid containing salt, sugar, urea and proteins. It is derived from amniotic cells by filtration or secretion. Fetal urine when kidneys start functioning, secretion of lung cells and placenta. So this amniotic fluid is formed from amniotic cells, fetal urine, secretion of lung and placenta, like that. And constituents of amniotic fluid. This contains metabolites and hormones, cells that are sloughed off from fetal lungs, placenta and amniotic sac and fetal urine. So this amniotic fluid is having, it contains the metabolites and hormones, cells that are sloughed off from fetal lungs, also cells that are sloughed off from placenta and amniotic sac and fetal urine. The functions of amniotic fluid: it permits symmetrical development and growth. It provides a water cushion to protect the developing embryo and fetus from jolts that mother may receive. It allows free fetal movements, an important factor for musculoskeletal development and blood flow. It helps to maintain consistent pressure and temperature. It forms hydrostatic bag and that helps in dilatation of cervix at the beginning of labor. So this amniotic sac, which is primarily between the epidermis and the amniotic and the trophoblast, as growing enormously, such that it is gradually covering the embryo, and then finally the yolk sac is drawn into this embryonic folds while embryo is folding, such that the whole of the coelomic cavity is obliterated by the growing amniotic cavity, such that the amniotic layer and the chorion, the outer layer of the trophoblast, outer layer of the coelomic cavity are coming together and they are thereby obliterating the coelomic cavity. So thus the amniotic cavity has now completely occupied the interior of the chorion. Now its functions is to give a pressure and to maintain the temperature. Then it is giving place for this embryo to develop. That means it is surrounded all the sides by fluid, no? So it can develop equally on all sides. And then it is protecting the embryo from jolts when the mother is all like jumping or something like that. This jolt will not reach the embryo because of this fluid present in the amniotic sac. And of course, at the time of parturition, this bag of fluid is coming first and it is dilating the cervix because of this fluid pressure. Then clinical aspect, amniocentesis, that is the drawing of amniotic fluid. It is a procedure in which amniotic fluid is aspirated from the amniotic cavity for diagnostic purpose. Why should we draw this amniotic fluid? To diagnose any congenital malformations or the sex of the baby and some of the development that is going. These things can be diagnosed from amniocentesis. When we are doing amniocentesis, that is during 14th to 15th week of pregnancy, and at that time the amniotic sac will be containing about 175 to 225 ml of fluid. So this is how we are doing amniocentesis. Actually, this should be done under ultrasonography. That means you are guided by the figure that is we are seeing in ultrasonography in the TVSP. Then only you can put a needle safely without touching the baby, just into the amniotic fluid. That means you have to pierce the myometrium, perimetrium, myometrium, and endometrium, and of course the amnion and chorion or the fetal layers. Then you reach this amniotic cavity. So of course here we are seeing how the amniotic cavity is gradually growing to cover the coelomic cavity and finally reach the chorion. See. This is the amniotic cavity which is enclosing the embryo. This gradually grows and completely covers this extraembryonic coelom such that it comes close to the chorion. So the amnion and chorion will be lying side by side as the embryo grows fully. So here you are seeing this is the amniotic layer and this is the chorionic layer, and both are coming very close to each other, and then they are present in the the uterine cavity, and the placenta is adherent to the. This one is the endometrium of the uterine cavity, and the uterus, and this is the only this part is the cavity because it is present in between the two endometrial layers of the two walls of the uterus. See, this one wall is bulging because of this embryo, and this other wall is just only having the endometrium. So in between you are having this the cavity of the uterus. Now this amniocentesis is done to examine the chromosomes in the cells of amniotic fluid for detection of genetic diseases. So after drawing this amniotic fluid, you draw cells, separate the cells, then you cultivate them, then you have to see the chromosomes present in these cells. Then you may find some defects in the chromosome to detect a genetic disease. Next, it detects the neural tube defects. So if the defection of neural tube is there, that can also be seen in the amniocentetic fluid cells. To detect the sex of fetus, of course, when you are seeing the number of chromosomes, you can see whether there are 2X chromosomes or XY chromosomes, so that you can determine the sex of the fetus. So you can see three different facts in this amniocentetic fluid. You can diagnose the sex of fetus, you can diagnose any neural defect present, you can diagnose any other genetic diseases that may be present. That is by the examining the amniotic cells. Now, the conditions vary. The amniotic fluid is less or more. If it is less, we call it as oligohydramnios. It is a clinical condition in which volume of amniotic fluid is less than the normal, which is about 700 to 1000 ml. So this amniotic fluid is 700 to 1000 ml normally, and if the volume is less than 400 ml or less, it is termed oligohydramnios. It is almost nearing the full. It occurs due to the placental insufficiency with reduced placental blood flow, agencies of kidneys, and loss of amniotic fluid due to premature rupture of amnion. So there are three causes that may cause this oligohydramnios. One thing is the placental insufficiency. That means less amount of blood is flowing to the placenta, and so there is less amount of amniotic fluid. And another is agencies of kidney. If kidneys are not produced, urine is not produced in the fetus. So again, urine is being added to the amniotic fluid, that is lessened. So there is a chance of oligohydramnios. Then loss of amniotic fluid due to premature rupture of amnion. If the amniotic layer is ruptured, fluid will go out. So also oligohydramnios, loss of fluid and leads to oligohydramnios. Then polyhydramnios. If the amniotic fluid is excess more than 2 liters in the amniotic cavity, we are calling it as polyhydramnios. This occurs due to esophageal atresia or defects of central nervous system, which may cause the fetus unable to swallow amniotic fluid. Consequently, fluid is not absorbed in the gut of the fetus. So, in polyhydramnios, you can diagnose esophageal atresia, whereas if it is oligohydramnios, it may be due to renal agenesis. So these are the two conditions of amniotic fluid which can be diagnosing certain important diseases in the embryo. So that's about the amniotic fluid, amniotic fluid. Now go for yolk sac. So as we have seen the blastocyst, the upper part that is between the epidermis and trophoblast, we have seen the amniotic cavity being developed, and the lower part between the endodermis and the trophoblast, we are seeing yolk sac. This yolk sac, an endodermal sac, develops from the cavity of the blastocyst. This cavity of blastocyst is converted into primary yolk sac when it is lined by flattened cells derived from the endoderm of embryoblast. So that endoderm of embryoblast is giving flat cells that are lining the inner wall of the trophoblast. So that there is a cavity found in between. This layer we are calling as Husser's membrane. Then this cavity that is present between the endoderm and this Husser's membrane, we are calling it as primary yolk sac. It is formed at the end of the second week. Then secondary yolk sac. With the appearance of extra-embryonic silo, the primary yolk sac becomes smaller and is termed secondary yolk sac. The cells lining yolk sac become cuboidal. Now the cells, Husser's cells which are flat, are becoming cuboidal and the sac is constricting, and this time we are calling as secondary yolk sac. Tertiary yolk sac, it is the remnant of secondary yolk sac. That means as the folds of embryo is occurring, we see that a part of the yolk sac is down into the folds, that is forming the gut of the fetus. So after the part that has gone in between the folds of the embryo, that is absorbed into the embryo, the part that is left over, we are calling it as the tertiary yolk sac. See, as the embryo folds, it takes up most of the yolk sac inside the body of the embryo to form primitive gut. The portion of the yolk sac not taken up inside the body of the embryo is termed tertiary or definitive yolk sac. It communicates with the midgut via vitello-intestinal tract. So the part of the yolk sac is absorbed into the embryo because of the folding of the embryo, and that part is becoming primitive gut. Whereas the remaining part of the yolk sac, that is remaining the outside this embryo, is still connected with this yolk sac, the part of the yolk sac that is inside. This connection we are calling as vitello-intestinal duct, that is connection between the gut and the yolk sac. So this is how the amniotic cavity, and of course you could say the embryo itself, embryo is folding on the primary yolk sac, so that a part of this secondary yolk sac, part of the secondary yolk sac is observed, is drawn into the body of the embryo, forming the gut, and the part remaining outside we are calling as the tertiary yolk sac, and this connection between this gut and the tertiary yolk sac we are calling as vitello-intestinal duct. This is vitello-intestinal duct. Of course, this is the gut that is formed of yolk sac only. This is the tertiary yolk sac that is remaining outside. And now the hind part of this yolk sac is forming a diverticulum, allantois. This allantois will talk. See, this allantois is going into the stalk of this embryo, and it will be finally forming the cloaca of this embryo. The other thing, this is the vitello-intestinal duct, this is the tertiary yolk sac, and of course secondary yolk sac, primary yolk sac we have already seen. The difference between the primary yolk sac and secondary yolk sac is the primary yolk sac is lined by Husser's cells which are flat, whereas the secondary yolk sac, after reduction of part of its size, it is lined by cuboidal cells, and it is called secondary yolk sac, which is extending into the stalk of the embryo as allantois. Functions of the yolk sac. So this yolk sac produces blood for embryo till the liver is formed in the first 6 weeks. Then formation of primitive gut. Its dorsal portion forms the primitive gut. Formation of allantois. A small diverticulum that arises in the caudal part of the yolk sac forms allantois. We'll talk allantois later. Now, Meckel's diverticulum. The stalk of the yolk sac usually detaches itself from the midgut by 6th week, and yolk sac gradually sinks as pregnancy advances. But sometimes if the stalk persists, it we are calling it as Meckel's diverticulum. So Meckel's diverticulum is almost persistent vitellointestinal duct. If this vitellointestinal duct stays back after completion of development, if it does not completely absorb, it is forming the Meckel's diverticulum. Allantois. So now allantois, which is formed from the hind part of the yolk sac. We are seeing it is a small diverticulum that arises from the caudal wall of the yolk sac on day 16 and extends into the connecting stalk. After the folding of embryo, the allantois is connected with the dilated terminal part of the hindgut called cloaca. So this after the folding of embryo, this allantois is connected to the dilated terminal part of hindgut, which is called cloaca. Now functions of allantois. It is vascularized by allantoid vessels that later become fetal umbilical arteries and veins. So these allantois are vascularized by vessels which finally become the vessels of the umbilical cord. Now in adults it is represented by median umbilical ligament. It also contributes to the formation of urinary bladder. This allantois is forming a part of urinary bladder, and it is represented by median umbilical ligament in the adults. Applet anatomy. The extraembryonic portion of allantois degenerates during second month of intrauterine life. The part of intraembryonic portion gets incorporated into the apex of the urinary bladder, and the remaining part evolves to form thick urinary tube called urachus. After birth, urachus becomes fibrous cord called median umbilical ligament, which extends from the apex of the urinary bladder to the umbilicus. The failure of urachus to obliterate leads to the formation of urachal cyst, sinus, or fistula. So now the extraembryonic portion of allantois degenerates during the second month of intrauterine. The part of the intraembryonic portion gets incorporated into the apex of the urinary bladder, and the remaining part evolves to form a thick urinary tube called urachus. So a part of this urachus, allantois, is incorporated in the apex of the urinary bladder, and the rest is involuting to form a urinary tube called urachus. After birth, the urachus becomes fibrous cord forming median umbilical ligament that connects the umbilicus to the apex of the bladder, which extends from the apex of the urinary bladder to the umbilicus. The failure of urachus to obliterate, if this urachus, which is tubed up, which is connecting the umbilicus to the urinary bladder, if it fails to obliterate, it will become a urachal cyst or sinus or fistula. Cyst is a encysted fluid, and that means a small cavity fully surrounded by wall. We are calling cyst with fluid. And sinus is a connection between the two cavities, and fistula is a connection between the cavity and outside the body. So that's about the urachus, allantois. Of course, urachus is also a large part of the allantois. Now, chorion. Chorion is highly specialized extraembryonic membrane that participates in the formation of placenta. It is formed by somatobularic layer of extraembryonic mesoderm and trophoblast. If you remember the class on the third week of development, there we have shown that how the extraembryonic coelom is formed from extraembryonic mesoderm, and then this coelom divides the coelomic mesoderm into a symptopluric layer and a splanchnopluric layer. This splanchnopluric layer only covers the primary yolk sac, whereas symptopluric layer covers the inside of the trophoblast as well as this amniotic cavity and extends it to the stalk of the embryo. So now this chorea, which is formed of symptopluric layer as well as the trophoblast, it is surrounding whole of the embryo. Now numerous small fingers-like projections arise from its surface called villi. So this outer surface of the chorea, which is formed by the trophoblast. Actually, it is formed by cytotrophoblast, outermost layer, and outside the cytotrophoblast, as cytotrophoblast divides, multiplies into cells, more number of cells, and here what is happening? Before the cell wall is formed, the cells are multiplying. So you are seeing a heap of nuclei with a protoplasm that we are calling as the syncytial trophoblast. This syncytial trophoblast is piercing the decidua of this uterus as finger-like projection, which we are calling villi. On the side of the deciduo capsularis, the chorionic villi regress and disappear, leaving smooth surface called chorion leve. So the side which is towards the cavity, we are calling as the deciduo capsularis. The villi there will regress and a smooth chorion leve is left. Whereas on the side of deciduo basalis, that is the chorion that is towards the decidua. We are calling it as decidua basalis, where the chorionic villi develop further and grow into decidua basalis to contribute to the fetal portion of the placenta. It is called chorion frondosum. Now, chorionic villi sampling. In this procedure, a small sample of chorionic tissue is removed under the guidance of ultrasound. So there also we are using ultrasound to get small amount of amniotic fluid. Here we are removing chorionic villi tissue under the guidance of ultrasound only, and here it is taken at the time of 10 to 11 weeks of gestation, whereas amniotic fluid is done at the 12 to 15 weeks of gestation. So here we are using this chorionic villi sample earlier to detect the genetic disorders. So it is useful, more useful than amniocentesis. That is, we are getting this chorionic villi sample earlier than the amniocentesis. Thereby, we are diagnosing the genetic disorders earlier than that we can diagnose the amniocentesis. Of course, it provides rich source of DNA, which is nothing but the chromatids, and where we can detect the abnormalities of the chromosomes. Now, implantation. Implantation is a process by which blastocyst is embedded in the endometrium of the uterus. It usually occurs during 6 to 7 days after fertilization. The intimate contact of fetal and maternal tissue occurs by implantation of blastocyst. The blastocyst surrounded by zona pellucida enters the uterus on the 4th day. The zona pellucida prevents blastocyst from sticking to the endometrium. As the blastocyst enlarges, how this enlarges? As it is entering the uterus, it is the fluid in the uterus is seeping into the blastocyst. We have already told, so only by just explaining. So as the fluid seeps into the uterus, blastocyst, the blastocyst is enlarging, and gradually as it enlarges, what is happening? The zona pellucida, which is a tight covering on the blastocyst, is broken up. So because of this stretch, the zona pellucida is broken up and ultimately disappears by day 5. So once this zona pellucida disappears, the trophoblast is exposed to the endometrium, that is the decidua of the uterus. So this trophoblast has the property of attaching itself to any tissue with which it comes in contact with. So till now, as the zona pellucida is covering this trophoblast, it is not able to stick to the uterine tube or the first part of the uterus, interstitial part of the uterus, and then now as it has entered the cavity of the uterus, by this time it has absorbed more fluid such that the blastocyst has grown more in size, and this zona pellucida is broken up and finally disappeared. Now the trophoblast cells are exposed to the uterine endometrium so that they can stick to the uterine endometrium, and from there these trophoblasts divide mitotically, forming syncytial trophoblast. This syncytial trophoblast invades the endometrium with the help of proteolytic enzymes secreted by its cells. The blastocyst goes deep until it is completely lies within the endometrium. The gap in the endometrium is closed by fibrocoagulum of the blood. So now it has completely dug the endometrium. The endometrium is prepared in the secretory phase of the uterus, and here what is happening? There are more glandular tissue, more connective tissue, and more fluid, such that it is very soft. That this blastocyst can go deep into the endometrium, and it is completely covered inside the endometrium, such that the gap is filled by the plug of coagulum of blood. So now it is inside the endometrial wall. So we are calling it as interstitial implantation. This type of implantation of blastocyst we are calling as interstitial implantation. The other types is eccentric implantation, where blastocyst is embedded in uterine crypts, that is in rats, and central implantation. In this type, blastocyst is implanted in the uterine cavity, that is in cows. Now, normal site of implantation. Where is this normal? The blastocyst is getting implanted normally. Normally, the blastocyst is implanted at the junction of the fundus and posterior wall of the body of uterus. Fundus is the topmost part, and posterior wall of uterus is below the fundus, body of the uterus. Fundus, body, and cervix, 3 regions. So fundus is the topmost part, and body of the uterus, that is to the posterior body of the uterus, at the junction of the fundus and the posterior wall of the uterus, body of the uterus. This implantation is occurring. But implantation anywhere in the upper part of the cavity is considered normal. So in the lateral part or anterior part of this uterine wall, just below this fundus, is considered normal. It should be above this cervix. Implanted concept produces a small swelling on the surface of endometrium. So it produces a little above the endometrial surface. Abnormal sites of implantation. These may be within the uterus or outside the uterus. So abnormal site of implantation within the uterus is in the lower uterine segment near the internal os. If this uterus has passed, actually the fundus is the part above this, the entry of the fallopian tubes. So as it comes out of the fallopian tube, it is exposed to the first part that is exposed is only body of the uterus. If it is exposed to the posterior wall of the body of uterus, there I told you by this time the trophoblast is free because the zona pellucida has dissolved. So now the trophoblast goes and attached to the posterior wall of the body of the uterus. That is the normal side. By chance, if this zona pellucida has not disappeared completely, so what is happening? This will pass through the body of the uterus, then this will get implanted at the cervix of the uterus. So this is the abnormal site in the cervix of the uterus. And sometimes, if this zona pellucida dissolves earlier, this may implant the ampulla itself or infundibulum a bit toward the uterus, and interstitial part of the fallopian tube. That means the part of the fallopian tube that is in the thick wall of the uterus, interstitial part of the fallopian. So these are the other three sides of the abnormal implantation in the tube. So we are seeing abnormal implantation in the uterus, that is the lower part of the lower segment of the uterus near the internal os. In the tube, it is the ampulla, the place where the fertilization is occurring, and the infundibulum is also there only, and the interstitial part of the frequent, interstitial part of the fallopian tube. So these are the other sites of the implantation, abnormal implantation. Then sometimes it comes out of this fallopian tube and gets inserted, implanted into the mesentery of the rectouterine pouch, or sometimes as the fertilization occurs in the ovary, before it comes out of the ovary, it itself develops into blastocyst and finally gets implanted in the ovary itself. So implantation in the abnormal sites, number one is in the lower segment of the uterus. Number two is in the tube, three parts. We are saying it is the ampulla, infundibulum and interstitial part. Number three is in the pelvic cavity, that is the mesentery of the rectouterine pouch, that is between the rectum and uterus, there is a pouch. So that peritoneal part is a site of implantation, and sometimes this may get implanted in the ovary itself before it is being expelled. That means the sperm has entered the ovary, it has fertilized the ovum there in the ovary itself, and there itself the blastocyst develops and implantation occurs before the expulsion of the ovum from the ovary. So here we are showing, this is the lower segment of the abdominal sides. No? This is the lower segment of the uterus. So this part is the fundus of the uterus, this part is the body of the uterus. So as soon as it comes out of this tube, by this time its zona pellucida has completely dissolved. This posterior part of the body of the uterus is the ideal site of implantation of the ideal. This part of posterior wall of the body of the uterus is the ideal site of the implantation of the blastocyst. Whereas if it has gone down, this is one of the abnormal site of implantation, where we are calling it as lower segment of uterus near the cervix, and the other is at the ampulla, the white part of this fallopian tube, or at the infundibulum, that is nearer its fimbrial end, or in the interstitial part, the part between the uterus, the part of the fallopian tube between the walls of the uterus, interstitial part. And sometimes it may go out and get implanted into the rectouterine segment, mesentery of the rectouterine pouch, and sometimes the sperm may fertilize this ovum inside the ovary itself, and there only the zygote develops and gets implanted in the ovary itself. So we are having the abnormal sites of implantation, the lower segment of the uterus, then the fallopian tube, the ampulla most common, then infundibulum, and then the interstitial part of this fallopian tube, and outside this we are seeing it is implanted into the mesentery, the rectouterine pouch, and sometimes in the ovary. So this ectopic pregnancy, that means the pregnancy in abnormal sites, usually do not develop in unfavorable locations. Here the endometrium will not be there, so there is no supply of food. So this ovum which has implanted, zygote which has implanted, will not develop, and it may lead to the death of the embryo and severe hemorrhage during the second month of pregnancy. So by chance, if the embryo has implanted in abnormal sites, because of the less amount of food, less amount of the nutrition, the embryo is going to die in the second month of pregnancy. And once it has died, there is a severe hemorrhage occurring because of this death of this embryo. Now, tubal pregnancy. The most common ectopic pregnancy is tubal pregnancy, and so that is a 95%, and of the tubal pregnancy, 80% of them are in ampullary region. So this tubal pregnancy in the ampullary region, here also it will develop up to the second month of pregnancy. There it dies and abortion can occur. Tubal abortion is choriodesicitual hemorrhage occurs around the conceptus which gets detached and expelled into the tubal lumen. It is often seen in the ampullary implantation. So in the ampullary implantation, there is a tubal abortion can occur because of the death of the conceptus and hemorrhage. Tubal rupture. If it happens to the implantation happens in the isthmus part or interstitial part, that is the very narrow part because it is passing through the uterine wall. No? So the rupture leads to the internal hemorrhage and patient goes into a state of shock because severe hemorrhage will be there, and because of the result of severe hemorrhage, the patient will go into severe shock. Tubal pregnancies are terminated through medical and surgical interventions. Not after rupture. If after rupture, it is only the surgical intervention that is needed in case of tubal pregnancy. And before that, there is a chance that a medical intervention can be done to remove this tubal pregnancy. Placenta previa. Normally, the blastocyst is implanted on the upper part of the posterior wall of the uterine cavity and the developed placenta is attached to the upper uterine segment. I told you the implantation usually occurs in the posterior wall of the body of uterus as soon as this the zygote comes out of the fallopian tubes into the uterine cavity. So that is almost the upper segment of the uterine, the body wall of the uterus. And if it is coming down, occasionally the blastocyst is implanted in the lower part of the uterine cavity near the internal os. Then developed placenta is attached to the lower uterine segment. It is termed placenta previa. So if this implantation is occurring in the lower part of the uterus near the cervix, and if the placenta develops there, we are calling it as placenta previa. So in the placenta previa, we are having first degree, second degree, third degree, and fourth degree. In the first degree, the placenta does not extend up to the internal os. That means it is attached to the lower part of the uterus, but the placenta is not approaching the internal os. The second degree, it is extending up to the internal os, but does not cover it. And third degree is it is extending, it's covering the internal os, but when os dilates during childbirth, the placenta is no longer occluding it. And in the fourth degree, placenta completely covers the internal os, and when the os is fully dilated during the childbirth also, this is still covering the internal os. So this type of placenta previa is termed central placenta previa, and this can cause severe bleeding after first trimester of pregnancy or during parturition. So this last type, that is the fourth degree, wherein the placenta completely covers the internal os, even the os is fully dilated. This type of placenta previa can lead to severe bleeding either in the first trimester itself or during parturition. So we are having this placenta previa because of the attachment of the zygote in the lower part of the uterus. So the placenta previa is having 4 degrees. First degree, it is attached to the lower part of the uterus, but it is not reaching the os of the cervix. And the second degree, it is reaching the os of cervix, but is not occluding. The third degree is it is reaching the os of the cervix, and it is not occluding the cervix when it is dilated. And when it is not dilated, it is almost occluding. But when during the parturition, as the os is dilating, this placenta is not occluding this opening. Whereas fourth degree, it is completely covering the os, internal os, and it is occluding even after the dilatation. So this type of fourth degree, of placenta previa is going to give rise to severe bleeding in the first trimester or during pregnancy. So this the fourth degree of placenta previa we are calling as central placenta previa. So placenta previa is almost 4 marks question. You have to just write these points in a good score. So now this figure also. So here the first figure we are showing, it is implanted, a part is implanted to the lower part of the uterus, suppose upper part also there is some and it is not reaching the os. A. Second one, it is reaching the os of the cervix, nearer. Compared to this one, it is very nearer, and still it is not blocking the os. Here in the third degree, it is blocking the os when it is not dilated, where once it is dilatation occurs, this will not block. And fourth degree, it is completely blocking the internal os, and even after dilatation also, the placenta will be blocking. So there will be severe bleeding during lactation, or sometimes in the first trimester itself, there will be bleeding and abortion can occur. So placenta previa, this figure is important. At the same time, you have to write the 4 degrees of placenta previa. Now deciduum. The blastocyst lies in the stratum compactum and bulges gradually in uterine cavity as development progresses. The portion of decidua at embryonic pole we are calling as decidua bassalis. The part covering embryonic pole, opposite embryonic pole, we are calling as decidua capsularis. And the rest that is lining the uterine cavity we are calling as decidua parietalis. The implantation provides nutrition to the embryo from the maternal blood, initially by diffusion and later through development of placenta. So here, this is the uterine myometrium, and this brown one, and then this yellow one is the uterine endometrium. Whereas here, the site of implantation, at the site of implantation, we are calling it as decidua bassalis. The endometrium at the site of implantation, we are calling it decidua bassalis. Whereas opposite to it, where it is covering the top of this blastocyst, we are calling it as decidua capsularis. The other part of the endometrium, that is covering the inner aspect of the uterus, we are calling it as decidua parietalis. These are decidua parietalis. So here again we are seeing decidua bassalis, decidua capsularis, and this part we are seeing decidua parietalis. Now umbilical cord. Umbilical cord is long, twisted, beaded cord about 52 centimeters, and like so it is a cord-like structure with beading appearance, and this is attaching the fetus to the uterine wall via the placenta. So the umbilical cord is connection between the fetus and the mother through placenta. It's about 52 centimeters, and it is a long twisted cord, and it is having beaded appearance. Beaded appearance is because the vessels, that is the umbilical arteries or vein, are longer than the length of the umbilical cord. So they are twisting inside this shorter umbilical cord, so beaded appearance is occurring. This connects the umbilicus of the fetus to the center of the fetal surface of placenta, which is covered by glistening amniotic membrane. The cord is twisted and present false knots. Length is about 50 to 55 centimeters. Breadth is 1 to 2 centimeters. Development: the umbilical cord develops from the connecting stalk, the part of the extra-embryonic mesoderm in which extra-embryonic column does not develop. Coelom does not develop. Initially, the mesodermal bar of connecting stalk is attached to the roof of the amniotic cavity at one end and trophoblast at the other end. Gradually, with further development of embryo, it becomes narrower and moves to the caudal end of the embryo. Later, with the formation of tail fold, it moves ventrally in the region of umbilicus. See, it is in the caudal end. As the tail fold occurs, it is coming into the ventral region, that is onto the abdominal wall in the region of the umbilicus. Now it connects the embryo with the chorion. As the placenta develops, connecting stalk connects the fetus with the placenta. Here, this is the connection stalk which is connected to the amniotic cavity and trophoblast, and this will in course of time develop into the umbilical cord. That mesodermal ball connecting stalk is attached to the roof of the amniotic cavity at one end and trophoblast at the other end. Just now I have shown. Gradually, with further development of embryo, it becomes narrower and moves to the caudal end of the embryo. So, as the amniotic cavity develops, it moves to the caudal end of the embryo. And with the tail fold coming, it moves ventrally in the region of the umbilicus. So, as the tail fold, this caudal end is coming into the center, center of the ventral part, ventral wall of the abdomen of the fetus. So now the umbilicus, it is in the umbilical region. Now it connects the embryo with the chorion. As the placenta develops, the connecting stalk connects the fetus with the placenta. The umbilical vessels develop in connecting stalk and primary mesoderm of connecting stalk undergoes mucoid degeneration. Umbilical vessels develop in the connecting stalk. So these umbilical vessels are developing in the allantois, and allantois is coming into the connecting stalk. So these vessels will be developing in the umbilical cord, and now the primary mesoderm connecting the stalk undergoes mucoid degeneration to form a gelatinous substance called Waters jelly. So the primary mesoderm is forming Waters jelly in this umbilical cord. The Waters jelly protects the umbilical vessels. As the amniotic cavity enlarges, it obliterates the extraembryonic coelom, which forms a tubular sleeve around the umbilical cord. This extraembryonic coelom is forming a sleeve, a cover over the umbilical cord. The beaded appearance of umbilical cord is due to twisting of umbilical vessels to accommodate themselves in the shorter umbilical cord. I told you at the beginning itself, the umbilical vessels are longer than the umbilical cord. So they are twisting inside the umbilical cord to accommodate themselves in the shorter umbilical cord. So here we are seeing a cross section of the umbilicus, wherein we are seeing an umbilical vein, umbilical arteries, and of course this is the remains of medullary intestinal tract, and this is the remains of allantois, and this Waters jelly is because of the dissolving of the mesoderm of the extraembryonic coelom, and the outer wall is the abdomen. So here what you have to notice is why vein is colored red and arteries are colored blue. Here what is happening in the fetus is the metabolites and the deoxygenated blood is coming through these arteries into the placenta, and from the placenta the oxygenated blood and blood with nutrients is going through the umbilical vein into the heart of the fetus. So usually the arteries are coming out from the heart and veins are reaching the heart. So here the vein is having pure blood because it is coming after purification in the fetus, and the arteries are having impure blood because the blood which the oxygen which has given off oxygen is going to the fetus, I mean the placenta. So now the placenta is just like lung. In the lung also we are seeing the pulmonary arteries are colored blue, whereas the pulmonary veins will be colored red. So because the oxygen has added in the blood in the lung and the blood returning from the lungs, we are seeing them as red. So now the contents of the umbilical cord. We have seen two umbilical arteries, one umbilical beam. Umbilical arteries will be always round, whereas umbilical beam is almost irregular shape, maybe oval. And then water jelly. I told you this water jelly is because of the disintegration of the mesodermal cells of the extraembryonic fluid. And remains of allantoid diverticulum. Allantoid diverticulum is extending into the umbilical cord and it is almost degenerating. Some part will be remaining, that is the remains of the allantoid diverticulum. Remains of vitellointestinal duct, yolk sac. Yolk sac is also similarly part of yolk sac is remaining in the stalk, so that will be there as a remnant of the vitellointestinal duct. Functions of umbilical cord. The umbilical artery is arising from the ventral division of internal iliac artery. Here, internal iliac artery of the fetus carry deoxygenated blood from the fetus to the placenta. After oxygenation in the placenta, the umbilical beam carries blood from the placenta to the fetus. Placenta is an organ of respiration in the fetus. Complication of umbilical cord. Cord prolapse. If the cord is long, it is likely to prolapse through the uterus during parturition, leading to a clinical condition called cord prolapse. In this condition, the cord is likely to be compressed between the fetal head and pelvic wall of the mother. This may lead to hypoxia of the fetus, which, if persists for more than 5 minutes, may damage the fetal brain. So, cord prolapse is the first complication of the umbilical cord. It is occurring rarely, but it is a complication. This cord prolapse occurs when the cord is very long. The cord passes through the cervix and the vagina cord out, and as the after coming head is coming out, it is compressing the cord between the head and the pelvic wall of the cord. So what happens when the cord is compressed? The blood supply to the fetus is obliterated. So the oxygen supply to the fetus is obliterated, and that may lead to the hypoxia of the fetus. If this hypoxia persists for more than 5 minutes, the fetal brain may be damaged. So, if the cord prolapse is there, you have to get the head out as soon as possible. You may do an episiotomy or you may do something to get the head faster. Now, coiling of the cord around the fetal head. In about 1/5 of cases of all deliveries, if the cord is long, it may encircle the neck and may cause strangulation. So, when something is encircling the neck, what is happening? The blood supply to the brain, and again, of course, the fetus there is no respiration, but it is causing strangulation, means that is constricting the vessels that are going to the brain. So, it also is going to cause the brain damage if it persists long. Premature separation of placenta. If this cord is short, if this umbilical cord is short, there is a chance that during parturition, it can pull the placenta before the fetus comes out completely. That is, premature separation of the placenta can occur if the cord is short. So, if the cord is long, there is a prolapse of the cord which can lead to the hypoxia of the fetus. And if the cord is long, there is a strangulation of the neck of the fetus if the cord turns around the neck of the fetus. And if the cord is short, there is a chance that the premature separation of placenta can occur. So that's how we have finished the umbilical cord, the allantoids, the chorea, the amnia, these things. So please read once or twice. These small chapters are very important for short notes, and you read and at the same time compare with the figures so that you can understand it. Thank you.